Hey there, Biology 12. As I said, I was going to go over a couple of these sample exam questions, so I'm kind of working off my iPad. I'll do a little scribbling, a little writing, a little yakking, and hopefully answer how some of these exam questions should go, so you can see the way to lay out the criteria for these. Nice thing is, I can pinch, zoom, write, yak, all the above, just like I do in class. Okay, so here we go. I'll try to go through as many of these as I can, but obviously I don't think I'll make it through all of them. So here we go. Explain the importance of the following organic compounds to cells. Okay, so what would glucose really be for? First thing, when you see glucose, you should be thinking energy, right? Glucose is an energy compound, and it's a monosaccharide. It's really what your brain runs on best. In fact, most of your cells would kill to just have a pure glucose source. And if you're ever in the hospital or you're ever in a bad way, they'll put you on basically on an IV, and they'll be pumping glucose into your system. So there you go. Your body doesn't have to break it down, because the glucose, this six-sided ring, at this point, is pretty well exactly what your body would want. So it's raring and ready to go. Proteins. Your muscles are primarily composed of this. And structurally, most of your body is too. And without it, you wouldn't have things like enzymes, which are little itty-bitty catalysts that make chemical reactions run in your body more easily. Here's an easy one. Insulin is an enzyme that's very good at taking excess sugar out of your blood and putting it into storage, into places like your liver and into your muscles, and where it stores it as, in animals, as glycogen. So it's an important thing. Steroids. Well, without steroids, we wouldn't have the base compounds for a lot of our hormones. So, we could say estrogen, estrogen, there we go, and progesterone in women. That's a pretty important one. I could label more. Progesterone, but I'll just run with this. Males, we'll put in the male equivalent here, testosterone. So steroids are important because they're little chemical messengers for our body, and without things like estrogen, progesterone, testosterone, our cells wouldn't and our tissues wouldn't take on certain new characteristics, like the ones we see when we hit puberty, for example. That's just, I'm putting these down because I think these are the best ones you know. All right, let's adjust that a little bit. There we go. See a little bit more. As far as fats go, they're probably the best energy source. I mean, that's why your body doesn't like to metabolize fat. Energy. Think of whales, insulation. Let's put down an extra one here. Padding the organs. And in some case, we create too much padding, right? If we get too overweight, that creates excessive pressure on the organs. That's not good for anybody. Okay? Phospholipids. These are the most important components of the cell membrane. So, I'll forego the cell and just write the term membrane. Without phospholipids, you wouldn't have this special little arrangement, which I showed in class, which divides the inside of the cell from the outside of the cell. This structure here. And on a quiz, it's pretty important. You should always put that in, because without phospholipids, we wouldn't have membranes, probably wouldn't have soap, amongst other things. They're pretty darn critical. Explain why each protein has its own physical and chemical identity. In a question like that, what you have to remember to do is to say that proteins are made up of amino acid sequences, okay, not just any amino acid sequences, right? So I'll have to say amino acid like that. There we go. And depending on the order of the amino acid sequences, we can start to go from a primary structure, okay, which is just sort of like a bead necklace, to a secondary structure, which would be like a helix or beta-pleated sheets. Silk is an example. Then we make globs, third degree. And then we move on to the superglobs, which are quaternary structures, fourth degree structures, which is why I'm writing fourth degree. That's not fourth to the power of 0, but fourth degree. And an example of a fourth degree structure would be something like hemoglobin, which carries oxygen, hemoglobin, and also something like insulin. These amazing proteinaceous structures that have really important jobs in our body. Okay? Okay. Chemical and physical properties of proteins. Well, usually when you think of a protein, you should think of it having an end like this, sort of this nitrogenous end. Okay? And sort of as you work your way through it, a protein itself will sort of have a carbon chain. Okay, doesn't really matter. I'll just draw a simplified one. And that hydrocarbon chain would be in between. But then they've got this interesting side group, which will relate with an R. There we go. And I just wanted to draw the other end. Now it is simplified because I haven't drawn all that many carbons in the middle. But you've got your side group here, which makes, depending on the length of it and depending on its construction, can make up different amino acids. You've got your carboxyl right here, and really you have sort of a nitrogenous end on the end here, sort of your amino end here. Think about it, it's not all that different when you look at nitrogen bonding with hydrogen. You're seeing something that kind of resembles, how can I put this? Well, NH3 is ammonia. So you're seeing a nitrogenous end and a carboxyl end. And they're very reactive. And in fact, the nitrogenous end and the carboxyl end will link end to end, and through the formation of water, kind of stitches them together. So chemically, this is what you would expect something like an amino acid to look like. Physically, it depends on what you've got for this remaining, this side group here. It could be phenylalanine, it could be arginine, it could be many different amino acids. That R group is all-consuming and all-important. In advanced biochemistry, you would go into that, but at the high school level, we just show you a few. Okay? Not too many. What do we got here? Moving on. Monosaccharides, disaccharides, polysaccharides. Okay, the easiest way to do that, I think, would be to draw something like glucose without putting in all the extra little bits. So if you think of glucose as being a more or less a carbon ring, like that, then what you've got is something, let's go over like this, there we go, that you can link up sort of end to end like this. There we go. Draw it as best I can. A little bit hard with the stylus, but you get the idea. So at this point, one glucose, you would have your monosaccharide, If we link two of them, let's say we just drew from here to here, you would have your disaccharide. And if you kept going, right, kept going over, then what you would have would be a polysaccharide. So I'll just say mono, di, and poly. So in the case of glucose, it starts out as glucose as a monosaccharide. Link it up twice. So if you've got glucose, glucose, then you've got a disaccharide. And in this case, that disaccharide is maltose. And if you keep linking up glucoses sort of ad nauseum like this, then really what you have is a polysaccharide. At that point, we say it's basically a starch. Okay? Remember that animal starch and plant starch are a little bit different. Plant starch is much more orderly, nice rows, and animal starch is a little bit more chaotic if you look at its structure. Not nearly so nicely laid out. Okay, question number 5. This is pay dirt right here. Draw a typical example of each of the following molecules. An amino acid. So what do? Well, you've seen me kind of draw one already. I don't expect on a quiz that you would draw all the types of amino acids. You would just have to know the fundamental structure. So look back at my example before, you're good to go. Monosaccharide, I think we just saw that, with something like glucose or fructose, right? Galactose. A triglyceride. At the high school level, what we get you to do is we just let this represent glycerol, which I'll put it with a G, and then what we draw is 3 sort of fatty acids off to the side, like this. That works out pretty well. You should know the structure of glycerol. So you should look at its carbon skeleton, and you should do the same with your fatty acids. Okay? In the case of a dipeptide, okay, let's take that down here. You should be able to take let's say we just do a quick one here, like this. And what you should be able to do is combine that with the nitrogenous end of another amino acid and realize that what you're going to get is the formation of water. Now, I don't have enough room, but with the removal of that, you should be able to look at a dipeptide and be able to draw this as it forms from here. In other words, the carbon, the oxygen, and the nitrogen, how do they link up? And that linkage, if we simply draw, we'll just bore a little bit of space over here, what you should see between the previous carboxyl group is you should see this, and that carbon leads to this nitrogen, and then that nitrogen leads off to the rest of the sort of the ensuing amino acids. So there's an amino acid this way, an amino acid this way. So what we've got, I call the con-H bond, is right here. And that tells you that you formed a peptide bond. And many of them, you could say polypeptide, you get the idea. A saturated fat. So what do we got here? I think we've got disaccharide covered. We can come back to glycerol in a second. But we really should hit a saturated fat and unsaturated fat and glycerol. One sec, I'm going to pause here and make a slide. Okay, so as far as that glycerol particle goes, glycerol is basically a 3-carbon structure. So let's get to a pen here. Switch up the color. Getting a little bored of that. Okay, so glycerol, really what you do is you link 3 carbons like this, and you realize that it's an alcohol. So this is the very reactive part. In fact, you're probably picking up on that by now. So there's your OH groups off to the side. Because carbon forms 4 bonds, you have to go like this. So you sort of think there's 1, 2, 3, 4. And look at this carbon. If you're in doubt, it's probably linking with hydrogen. There you go. Go like that, go like that, and there we go. So some people have called this glycerin, but got to remember this is glycerol, and it's because of these hydroxide groups on the side. Okay, so you should be able to draw it just like this, okay? Go to our next slide. Something like a saturated fat. Okay, by saturated, we have to be very clear what they're talking about here. When they're talking about a saturated fat, they're talking about saturated for what? What's it full of? Saturated. They're saturated by hydrogen. Now, these are the kind of fats, like the animal fats, that are very unhealthy for us. They'll be solids kind of at room temperature. So if we look at something that's saturated, really what we've got with a lot of these fats, I'll just draw a carbon chain here. And it's just saturated with hydrogen. Imagine the maximum amount of hydrogen you could. I mean, if carbon could pull it off, it will share with 4 hydrogens. I mean, you look at methane, right? Hydrogen's an excellent electron sharer. Oxygen figured out that trick with water. And here we go. And we'll just draw sort of the final, we'll draw a reactive end here. There we go. Carboxyl group there off the end. COOH. Now that's saturated, so you will not see any double bonds really between any of these carbons. Nada, zip zilch. Now when we look at a saturated, or an unsaturated fat, so let's say we've got the same, I'm just drawing random carbons here. And what we'll do this time, let's throw a CH3 off the end like that. There we go. Let's say instead what we had is we had a double bond here. So in this case, hydrogen sort of needs, think of it as not quite 4 bonds, but it needs to share 4 electrons to become electronically stable. In this case, it didn't need that extra hydrogen down here. Let's throw another one over here. This would fulfill this carbon. This one here needs hydrogen. This fella here, he only needed 1. But you notice that if there isn't a double bond, that carbon will form up with more hydrogen, and that's exactly what we're talking about here. So let's put the carboxyl group off the end. Not perfectly drawn, but it'll do. So this is what makes it unsaturated. It doesn't have as many bonds with hydrogen as it otherwise would, because it's double bonding with, it's playing sharesies with an adjacent carbon. Okay? These kind of fats tend to be liquidy. So a lot of unsaturated fats, canola oil is like this, right? A lot of vegetable oils will do this. They tend to be healthier for us. I always thought, imagine if bacon fat was at room temperature in your arteries. You wouldn't really survive very long. Saturated fat is very bad for you, and it's an animal fat. And if you don't believe me, cook bacon, let it cool down, and look at it in the pan. It's just solid as a rock. Well, solid and mushy anyways.